Okay, a fine day, folks. Welcome. Thank you so much. You know, the presents that you give are your presents. So just being here is a present to everybody right next to you. I just want to say, don't be shy. If you need to ask somebody if they have something that you could use, please do. This is a community. And other than that, we're here for you and you're here for us. This is a together game and we're in it together. Over to you, James.
Thank you, McFinn. We've got another wonderful show today. Before we jump into everything, I'm going to turn it over to Mira for our little announcements.
Hey, everyone. It's so nice to see all of you again today. I just have a couple of announcements, still similar to the last week, but we are really still pushing both of our studies. We are doing some really great work. So one of them is our speech study where we are collecting voice samples for about 15-minute sessions once a month. It's a very low ask, minimum time requirement. We can help walk you through the steps. Everything is all easy to follow on our online format. However, we can also personally help assist you through that. Your speech is really important, and that's simply because our data scientists are finding that we're finding some cues that might hint towards some digital biomarkers, and we're hoping to get our research moving. So that's the speech study. The other study that I've been talking about, we are really excited to let you know that we've been launching it. It's the Radcliffe study. It's multimodal. We'll be collecting PFTs, that's your lung function test, your walking ability, and your speech analysis. And those will be completely remote, which means at the comfort of your own home. And in order to be in this study, you'll talk to me. You do need to be able to walk. So we are aiming for early diagnosis. However, we are still accepting slow progressors. But if you're interested or have questions, please fill your name out on our www.everythingals.org/research. James helped me create a YouTube video. So now we are live on YouTube, where I talk to you a little bit more about the Radcliffe study. And so just fill out your information and we'll contact you. We're really excited to get started. If you have any questions, please send us your email. We're happy to get in touch with you. I don't want to take too much time away from Merritt and Nazem. I have had the pleasure of meeting them both in person, and they are just wonderful human beings. And I'm so excited that they get to share their time with you and tell you really exciting events. So I'm going to hand it back to James to do more of a formal intro, and then we'll get started. Let us know if you have any questions.
Perfect. Let's just jump right into it. I'm going to introduce our guest speakers for the evening, starting with Dr. Merit Cudkowicz, who is the director of the Sean M. Healey and AMG Center for ALS, chief of neurology at Mass General, director, and the Julieanne Dorn Professor of Neurology at Harvard Medical School. Dr. Cudkowicz's research and clinical activities are dedicated to the study and treatment of people with amyotrophic lateral sclerosis. She is one of the founders and past co-chairs of the Northeast ALS Consortium and is launching the first platform trial initiative in ALS, the HEALEY ALS Platform Trial, a program that will greatly accelerate therapy development in ALS. She is joined today with Dr. Nazem Atassi, who is the global head of early neurodevelopment at Sanofi, where he oversees the company's early clinical development portfolio in neuroscience. He completed his master's in medical sciences at Harvard Medical School, neurology training at Boston Medical Center, fellowship in neuromuscular disorders at Mass General Hospital, and obtained his MBA at MIT. He continues to attend clinic at Mass General Hospital, where he cares for ALS patients and their families. Between the two of them, we've got quite the brain, so I'd like to welcome our two guest speakers for the evening.
Thank you very much for inviting us here. It's always great to be here, and it's really fun to be here with Dr. Atassi, who I've worked together with for many, many years. I've lost count, and who's really dedicated to developing treatments for people with ALS. I was, before Nazem starts, I want to say that today I got an email from Judith Carey, who's our research access nurse, who told me that getting lots and lots of calls for enrollment in neuroscience. And the reason is because neuroscience recently spoke at Everything ALS and got the word out about their exciting trial. So it really shows the power of this amazing group to help, help recruitment so that we can get answers to whether a drug works or not faster. So thank you for that.
Thank you, Merit. That was about 7 days ago, exactly. So thank you.
Fantastic. So is Nazem on the call? I saw him before.
Yeah,
great.
I'm here. Thank you for the introduction. And, as Merit said, I think the best thing on both our CVs or our bios is that we work together, Merit and I. Merit was and still continues to be my mentor. She trained me when I was at Mass General, and we worked together for 10 years, and we continue to work together. And I feel very privileged to be able to continue to working with her. I also want to thank Everything ALS for organizing this, for all their efforts, really. It's Indu and Mira and everyone at Everything ALS. It's really amazing what you were able to create. And thank you for hosting us. Really grateful for all of you. And thank you all for joining. I know for some of you it's late at night, and some of you it's difficult to join. So I really appreciate joining this call.
Great. So I think we'll jump in. I think Nazem, are you going to share the slides?
Sure. Yep. Let me do that right now.
And you should have access to do so. Perfect.
Great.
Do you see the full slide?
Yeah, you
We
might
see
be in that weird thing where we see your next slides. You know, to change this
Oh.
way.
Yeah, if you just go up there to use slideshow, I believe, there you go,
There
perfect.
you go. Yeah.
Is it better?
Yeah,
Yes.
perfect.
Perfect. Okay. So maybe I'll start, and then I'll turn it over to Merit. I'm just gonna move this control thing. Okay. Perfect. Let me do this. Okay, fantastic. Okay, can everybody hear me and see me and see the slides?
Yes.
All good.
Perfect. Okay, fantastic. Okay, so today we're going to talk about the ALS Himalaya trial. This is a phase 2 trial. It's multi-center. It's a randomized. That means people have, by chance, being able to be on a drug or a placebo. It's double-blind. And it's followed by an open label long-term extension. So everyone who completes the study will be able to take the active drug afterwards. And really, we're trying to evaluate the efficacy and safety of our compound. Currently, it has a number. Hopefully soon we'll have a name. It's SAR443820 in people with ALS. And I'll tell you a little bit about the mechanism of action and how we think this compound might work. And then Merit will tell us about the trial design and the recruitment and the sites and all of that. So this is just kind of a brief overview of this program or this compound. So the mechanism of action is summarized by that this is the CNS penetrant. That means this compound gets to the brain, and it's a selective small molecule. It's an inhibitor of RIPK1, and I'll tell you what RIPK1 is, and it's a relatively novel pathway in ALS. RIPK1 pathway includes both apoptosis and necroptosis, and this is basically technical terms for inflammatory cell death. So regular cell death and inflammatory cell death that is related to inflammation. And I'll tell you a little bit more about that. It's an oral tablet, so that's kind of important. The route of administration is taken twice daily. So it's a pill. And currently we are in phase 2. So we have already completed first-in-human studies. I'll share some of the results from the first-in-human studies, and then Merit will tell us about the phase 2 trial. So what is RIPK1 and why we want to inhibit RIPK1 in ALS? So RIPK1 is a key target really at the intersection of inflammation and cell death. And these are two phenomena that we have seen over and over in ALS. The motor neurons die and then there's inflammation around these motor neurons. So this is a cartoon basically over here, and what you see in this cartoon, you see this this cell. This is a neuron, a nerve cell in the brain, and this is the pathway. It's RIPK1, and it can bind to either these capsase and cause apoptosis, which is cell death, or it can bind to other kind of complexes, and it can cause this inflammatory cell death. And what happens is that the neuron dies, and then when it dies, the remnants are these kind of damps, we call them. And then what happens is that after the neuron dies, these damps activate the inflammatory or reactive cells in the brain, mainly the microglia. And these cells secrete more pro-inflammatory compounds, which in turn activate this RIPK1 pathway again. So as you can see, there is a vicious cycle of activation of this pathway leading to cell death, and then that cell death leading to activation of the same pathway in different type of cells, causing more inflammation and causing more cell death. And the goal here is really to break this vicious cycle, hopefully to reduce both inflammation and to reduce the death of these cells, the motor neurons. So what is the link to ALS? How do we know that this target is actually important in ALS? And there are different clues that we looked for and we found. First, we have what we call a pathological link. That means that we looked in pathology under the microscope at different samples from people who passed away from ALS. We looked at their spinal cords. And what we saw is that there is increase in RIPK1 and the phosphorylated component of RIPK1 in people with ALS, in the spinal cords of people with ALS compared to the healthy controls. And this is, each one of these dots actually is a sample from a patient. And you could see in ALS, there is an increase in activation of this pathway, as we can see it from samples from patients with sporadic ALS. In addition to the pathological link, there is a genetic link for RIPK1 with ALS. It's not a genetic link in terms of RIPK1 itself, loss of RIPK1 or activation of RIPK1 causes ALS. It's actually more that there are a number of genes that are implicated in ALS, and they are highlighted over here. They're different genes, and they encode different proteins, but in every single case of these genes that are implicated in ALS, there is a activation of this pathway of RIPK1. So if you remember that slide I showed before, how there is this increase of RIPK1, it's happening when you knock down, or when you actually take out, these ALS genes, you see the activation of RIPK1. So that's a genetic link, not only to one, but a number of genes that are involved in ALS. So then we said, okay, there is a pathological link from samples from patients who passed away. There's an activation of this pathway, and there's a genetic link. Let's try to test modifying this target, the RIPK1 target, in animal models of ALS. So this was done actually both internally at Sanofi, but also was done externally. So independent labs have done similar experiments, and they all reached, and many of these is published, had reached similar conclusions, that if you treat these mice with compounds that inhibit RIPK1 around day 70, so before they develop the disease, or around the time when they develop the disease, not very early, not when these mice are very young, but around the time really when they develop the symptoms, there is a delay in the symptom onset in the mice treated with the RIPK1 inhibitor compared to mice that are untreated. And this, as I said, was replicated both externally and internally, we did the experiment with different backgrounds of these SOD1 mice. These are ALS mouse models. And we saw the same thing. And also we saw preservation of weight as well in these mice. And then we looked at the function of these mice, not only time when they had the first symptom, but also the function that they had. And when we looked at different type of measurement of function in mice, for example, the wire hang, which is how long the mice can hold on to a wire. The distance traveled, this is you put a mice in open field and then you see how far they go in that open field. Movement speed is how fast they go in that open field. And then lastly, we looked at hind limb strength. The mice sometimes try to kind of stand on their hind limb, like the legs basically. And you can see how strong that hind limb. And in every measure of these, these are representative of muscle strength and muscle function and activity. In each case, the mice who are treated with the RIPK1 inhibitor, so you can see them here in blue, did better than the mice that they were not treated with the RIPK1 inhibitor. And then we developed a compound. So all of these experiments that were done in mice using kind of tool compounds. But then we developed an actual compound. This is the SAR 443820, which is a RIPK1 inhibitor that we can test and take to humans. So we did first in human studies to look at the safety and look at the different parameters of how long the drug stays in the system and how long it takes to get excreted. And then also we measured how much of the drug gets to the CSF, the spinal fluid, which is very important in ALS. You want to be sure that the drug that you take actually gets to where it should go, to the brain. So we did what we call first in human studies. This is single ascending dose. That means each one of these healthy volunteers took one dose of the compound. And then we measured all of these parameters. And then here you see this in this little box is the people who took, about 12 people who took this compound, the RIPK1 inhibitor, and we were able to measure the levels in the CSF, in the spinal fluid. So these people had lumbar punctures to measure the amount of drug in the CSF. And then after that, we did multiple ascending dose. So that means we did dosing for up to 2 weeks of different dosages. And what we found is that overall the compound was safe, well tolerated. We had about 116 healthy volunteers who joined the studies. However, this is in healthy volunteers, was safe and well tolerated in healthy volunteers. The safety and efficacy is now being tested in the Himalaya trial, which Merit will tell us about. We also actually were very happy to see that actually the compound is fully CNS or penetrant to the central nervous system. The ratio of the levels that we saw in the blood or in the plasma compared to the levels of the drug that we saw in the spinal fluid were 1-to-1. That means the blood is the compound is fully penetrable. It can get to the spinal fluid freely. As I said, it's very important for treatments that targeting diseases like ALS. And finally, we were able to measure how much inhibition of this target we have. And we saw really very high inhibition, especially at the highest dosages that we gave, but really across most of the dosages that we tested, we saw high levels of inhibition of this target. That means we have a very potent compound, which is also important that we not only been able to show the safety within the healthy volunteers, that it's goes to the brain, to where the action is, but also to show that actually the dosages that we are taking to the Himalaya trial, to the phase 2 trial, are actually potent and strong dosages that actually hit the target really hard in order to be able to test if this compound works or not. So that's kind of the scientific mechanism of action, background, why RIPK1 inhibition, and the experience with first-in-human studies. And now I'll turn it over to Merit to tell us about the Himalaya trial, the phase 2 trial in ALS.
Thank you, Nazem. So the Himalaya trial is now launched. This is a phase 2 trial to look at longer term safety as well as efficacy of this drug in people with ALS. And I'll start by saying that Sanofi really listened to the Sandy Moore's principles of really engaging people living with the ALS in the design of this trial. So I'm very thankful to them for doing that. So the goal is to look at efficacy and safety. It's a phase 2, which means that we, as you heard from Nazem, the phase 1 really showed the dose, determined the dose that was needed for maximal RIPK1 inhibition, showed some preliminary safety. But this next phase 2 study is really the first one to look at efficacy and longer term safety. The design is, what I would say, is very people-centric. So we're really trying to answer the key questions, but really minimizing the number of people on placebo, minimizing the duration of time on placebo. So there's 2 parts to it, part A and part B. We're looking for 260 participants. They will be randomized 2-to-1 to active drug versus placebo for 24 weeks. And after 24 weeks, everybody, if they finish the 24 weeks, can go into the long-term extension, which is the part B. And that will go on for probably about a year and a half, really until we have the results of the part A. The main outcome in part A is to see the effect on the change in ALSFRS. In part B, we're also going to be looking at the effects on this measurement called CAFS, which looks at both function and survival. We're going to look at much more than the ALSFRS. We're going to look at breathing. We're going to look at muscle strength, quality of life, some fluid biomarkers, including serum neurofilament, safety, tolerability, some PK or pharmacokinetics of the drug. And we're also added something you guys know a lot about, which is a digital speech analysis in the study. Next slide, please. The criteria are really designed to be able to get an answer in the 6-month double-blind period of whether the drug has efficacy or not and safety. So it's a broad range for age, also of where people are in the illness, including people with these different criteria called possible, clinical probable, probable laboratory supporting. What this really means is the number of regions in the body that are affected by the ALS. It is looking for people who are earlier in the illness, so less than 2 years from first symptom, and the symptom really defined by motor weakness, breathing more than 60%, able to swallow the study tablets at the screening visit. The study drug is crushable and it can be given into a G-tube during the study. And people can be on riluzole and they can be on edaravone, though we really ask that people are on a stable dose of those if they are on it at the beginning. There are a few drugs that can't be taken with that, and we can go into a little bit more detail what those are, but there are drugs that are that inhibit or induce this particular pathway called CYP3A4, which can either change the concentration of different drugs, either increasing them or decreasing them. Anyone with a history of seizures, which again is not very common in people with ALS, cognitive impairment, and there we mean really someone that is not able to understand the protocol or give informed consent. And then people in other clinical trials for ALS are taking investigational treatments. Next slide, please. This is the schedule of the visits in the part A, which is the double-blind period. And so there's what we call a screening visit where people come in and we see if someone's eligible for the study. And if they are, then they come in and they start the dose. There's a visit at 2 weeks and then 4 weeks, 8 weeks, and then it becomes less frequent after those first first visits. And that's pretty common in these phase 2 studies, that we do more safety assessments early on, and then it becomes less frequent. In the part B, which is where everybody's on the drug, again, we have some more frequent visits initially, and then a little less frequent. And most of those visits are at the site, but they're less frequent. Next slide, please. So this is a global study. So there's study sites really all over the world, in Europe and in Asia. In the United States, there's 19 sites that are listed here. These are also found on the clinicaltrial.gov website. I think there's a lot of flexibility if someone doesn't live near a site to help with travel and help people participate if they would like to be in this study. Next slide. The study is going well. It started in Europe first, last April. It started more recently in the United States, November, and as of November 10th, there's 51 people who are in the study. We're looking for 261. So this is really at the beginning stages of enrollment. But now that many of the sites are open, the hope is that this will be enrolling quickly. Next slide, please. We are actively looking for people interested to be part of the study. Sanofi has set up a website which has a lot of information about the trial, and can help people find a site near them. The next slide, I just want to repeat that really from day one, this is a study that had a lot of input from people living with ALS. We're very grateful for those people who gave advice to Sanofi and to the steering committee about how to design the study in a really patient-centered manner. So with that, I hope we have a lot of time for questions. I know there's a few that have come in.
We got lots of time for questions, which is great. I'm going to start it off, and then Grace, who is another Everything ALS fellow, will be alternating them with me. If there's one of you that is more keen to answer it, feel free to jump in. But the first question, and I hope I'm reading this one correctly, can one with OPTN participate in this trial?
Yes, absolutely. So that's optineurin, and that there's no exclusion for by a particular gene that someone might have.
Yeah, that is correct. And actually, a lot of the initial genetic links and the work on this pathway was done in optineurin models and samples with optineurin. So yeah, absolutely.
Hi, everybody. I'm Grace. So the next question, kind of around the same idea we have, has this been tested on anyone with sporadic non-genetic ALS?
Yeah, so this is the first study in people with ALS. So the phase 1 was done in people without any illness. So this is, this is the first one, but the biology for this makes absolute sense for people with both the genetic and the non-genetic form of the illness.
We love a trial that includes both, so thank you for that. Moving on with the exclusion and inclusion criteria, can a person on open extension trailose also participate in this trial?
So unfortunately, no. So trailose, because it's an experimental drug, it's not FDA approved yet for ALS, it would be exclusionary. So if somebody wanted to be in this study, they would have to stop trailose for 30 days or 5 times the half-life, whichever is longer. And I think it would be 30 days for that. But that would be definitely something you want to discuss with your, with your neurologist to see what might be the best option for you. I'm only answering these questions because I know there's some hard ones coming in that way, and Nazem can get the harder ones.
Well, we have another question up involving exclusion criteria. How is cognitive impairment for exclusion determined?
I'll ask Nazem maybe. Nazem, do you want to start with that one?
Yeah, of course. So it is exactly as Merit mentioned. It's really excluding people who have clear frontotemporal dementia. These are we know there is an overlap between a small overlap between ALS and frontotemporal dementia. There's a small portion of patients who have both. And the idea here is to have people who have the capacity to make a consent, basically, to join the study. So this is something that, you know, the site investigator who is enrolling in the study will know.
So it's more of a clear-cut observation than a proper diagnosis, I guess, is the correct wording.
Usually it's clear if someone has dementia or not. For a neurologist, usually it's clear, and for patients. And it's a very small percentage, actually, of ALS patients who have that. So
yeah. We often we definitely leave it to the site investigator, but often site investigators will ask the participants certain questions about the trial to get a sense of their understanding of the procedures and ability to give informed consent.
Perfect. I have a feeling this next question is going to have the same answer as the Trehalose one, but I'm going to ask it anyway. If a patient is on Relyvrio, AMX 0035, are they qualified to be in this trial as well or not?
So the answer is slightly different, actually, for this one. So as most of you know, Relyvrio was just approved in the US, in the United States. And this happened while our study is enrolling. So we are actually in the process to consider allowing people who are on stable dose of Relyvrio to enter the study. So whoever wants to enroll in the study, before enrolling in the study, they need to be on a stable dose of Relyvrio before enrolling. We still don't have that approval. This needs to be approved by, you know, different institutional review boards and the FDA and, you know, and health authorities and all of that. So we haven't gone through all of that, but the intent is to hopefully allow people, just like we allow people to enter the trial on Riluzole, we allow people to enter the trial on stable dosages of Radicava, the two approved medications in the US. We're trying to do the same for Relyvrio.
It is a little bit of a different answer. Thank you.
The difference is actually because Relyvrio is not considered experimental in the US versus, you know, the question was about an experimental treatment.
Yeah.
The initial question with telehost.
Well, great. Thank you for that answer. The next question that we have here is, would travel expenses be covered for participants if they weren't close to a center?
Yes, the answer is yes.
Short and simple. Is there an EAP for this trial?
So EAP is Expanded Access Program. And there is not currently an Expanded Access Program. Currently, we have an open label extension, which would allow all the participants in this study to have access to the active treatment after 6 months of double-blind period. If the results of the first part, the 6-month part, are positive, then this is when we would consider having an expanded access based on a positive results. Currently, this is a compound that, you know, although as I've shown you, there has a lot of links to ALS. The mechanism makes sense. We have mouse studies. We have safety from first of humans. We don't have any data on efficacy in people with ALS. So once we have that data, and with enrollment, you know, and joining the study, you'll help us to get to that answer quickly. And then this is when we consider having an expanded access program.
Perfect.
All right. So the next question is, are fast progressor candidates for this trial? And do you plan on later stages? If so, will it be sooner or later after the 2A/B studies?
I can answer maybe the faster question, then maybe Nazem, you can answer about future plans. So the inclusion criteria don't necessarily enrich for faster participants, because with the less than 2 years, it's also taking people with a broad range of the illness. So some people with possible, which means one region involved, and some with definite, which is 3 regions involved. So there'll be some heterogeneity, as opposed to, for example, the Radicava and the amyloid study, which took people in the first 18 months with what we call widespread illness, definite. So I think here is a broader population already in this study, but people earlier in the illness than perhaps a study that would go out to 3 years. So I don't know, Nazem, if you want to comment about future studies or plans.
Yeah, so we're definitely committed to ALS and to drug development in ALS. Obviously, this question depends on the results of this current trial. If you notice, this is a relatively large phase 2 trial. This is not a phase 2 trial in 20, 30, 50 patients. This is a phase 2 trial in 261 patients. So it's getting close to a phase 3 trial, if you want to call it. So really depends on the results of this study, if they're positive and how positive. We will decide the next steps afterwards. For example, if it's very positive, that to the point that leads to approval, then the drug will be available and approved for everyone. If there's requirement to replicate or to do larger studies based on the results, we will take it to the finish line.
I think you hit a key point there in stating that this is a larger participant-involved trial, which is always a huge thing we like to hear. Speaking of the trial in general, did you or is there going to be stratification for bulbar versus other presentations of ALS? I think
this is broad, and that people could be in who are bulbar onset or limb onset, and both are included. And we'll certainly do analysis in subgroups based on site of onset.
Okay, our next question here is the drug available through expanded access, and is it being considered for inclusion in the HEALEY trial?
So the majority of companies, kind of like Sanofi, will consider expanded access after they have initial, some initial data of the safety of the drug and efficacy. So I think it sounds like the plan for Sanofi would be, after this study, depending on the results for the expanded access. I will confess that I did try to convince Sanofi to join the platform trial, but not every drug company has to join the platform trial. There's there's a need for many types of designs, and this is a global study, and so the platform trial is really a US-based trial, and so there's advantages to going global as well, because you can perhaps enroll faster. You can also get a wider group of participants, and it helps with regulatory improvement elsewhere. So anyway, and this is also taking people slightly earlier in the illness than the platform trial.
Maybe I want to put a plug for the platform trial here. We really thought about it very seriously to join the platform. I think it's the most efficient way really to test drugs today in ALS. It's a very robust, it has very robust features in terms of the design and the statistics and the all the kind of details that goes in the trial. It's complex, it's not simple, and so I really commend Merit and the team to pull it all together. It's not easy at all, and actually it does accelerate the development, not because it's very efficient, not only because it's very efficient, because also it attracts a lot of companies to actually, who are not thinking about ALS, to actually think about ALS. So, I can keep going on and on. I'm a very big fan of the platform trial. I think for us, it was really the global piece that we wanted to do a study that, covers. I think you saw the map. the study is enrolling in China, is enrolling in Japan, is enrolling in many countries in Europe, in Canada, and in the US. So I think that's mainly the reason why we didn't join, but it's always on our mind, the platform trial, maybe hopefully for future programs.
You actually answered one of the questions that was coming up, so I love when that happens, so thank you for that. There's an interesting question here next. Is there a patient caregiver advisory board for Sanofi that gave the input, or was it obtained another way?
Yeah, so we met with a number of patients and patient advocates and foundations, and we got feedback on the protocol, on the things that we can improve with the protocol. We also have an amazing steering committee. Merit is the chair of the steering committee, with people who have a lot of experience helping people with ALS. And we actually made a lot of changes to this protocol. If you look at the initial kind of thoughts and designs of this protocol, we increased the ratio of people on a drug from being 50-50 to 2-to-1. So there's like 2 chances of being on a drug compared, to 1 chance of being on placebo. So it's a big change. It basically means that we're going to need more patients in the trial, but that was fine because we wanted to be sure that this is a patient-friendly study. We were thinking of a longer placebo-controlled period, a year, and we heard the patients telling us that 1 year is too long. So it is also a kind of a compromise for us. So we made it a 6-month double-blind study. We also heard the patient telling us how important it is to have access to the active drug after they complete the 6-month period. And we made the part B, which is the open label extension, part of the same protocol. So it's not another consent form and another protocol that you qualify or not for. It's part of the trial. You finish the 6-month and then you just continue taking the active ingredient, the active drug afterwards. The number of visits, the phone visits, the travel help with the travel, all of these things are things that we heard the patients and we tried as much as possible to accommodate and adjust the trial design, again with a lot of advice from Merit and the steering committee of the trial.
Great. Well, thank you guys for that answer. So the next question is, with your first human studies, were there any side effects that you guys noticed? Anything important?
Nothing really that we think is related to the compound. So it was relatively safe and well tolerated in the first human studies.
Next question is a little in depth, so bear with me. There's always seems to be confusion if a drug is crossing CSF or BBB. The current drug you said is concentrating on CSF, but do you think it's also maybe crossing BBB to get into the central nervous system?
Yeah, that's a very good question. It seems like a scientist asked this question. It's a very technical question. So we can only measure drug in this spinal fluid. We cannot measure it in the brain in living humans because that means a brain biopsy, which we don't do. But we know from the animals that if you have drug in the spinal fluid, the drug actually goes to the brain tissue. We measure both in the animals. So we know that if we see the drug in the spinal fluid, that means we know that the drug actually will make it to the brain tissue itself. So in the animals, we have that data. In humans, obviously, we don't.
So our next question is also a little in depth, and it says, as you said, the drug is highly susceptible to CYP metabolism. So the patient will not be under CYP inducer and inhibitor. Does the drug undergo the liver first, and is it susceptible to CYP of liver enzymes?
Yeah, I'll take this one. So yeah, the most oral drugs, they get absorbed in the GI system, in the stomach and the GI system, and they go to the liver first, and then they get distributed first pass. So this drug actually has a relatively low interaction with other drugs. So only strong inducers or inhibitors are of this. These are basically the enzymes that metabolize a lot of stuff in our body. So only strong inhibitors or inducers interact basically with this drug. And we have a list in the protocol of these drugs. Nobody is able to memorize that list. So we have them in the protocol for investigators. But we did look at the common medications that people with ALS would take. And actually, none of the common medications that ALS patients take belong to that list of prohibited medications. So unless you are taking some medication for a very different reason, a very different disease, usually these are not the common medications that we all that a lot of people take.
Excellent. I know this was focused primarily on ALS patients, but can a patient also be PLS? And if so, do you feel this drug may be suitable for PLS patients in the future?
Such a good question.
It is a good question, yeah.
Yeah, so in this trial, it's for people with ALS, but I really do think we need to figure out how to do trials with PLS, and this is actually something Nazem was working on when he was in academia. I do think that some of the biology is overlapping. The challenge we have in PLS is that we don't have good cell or animal models to really work it out like we do in ALS, and we lack kind of some of those tools of how to do trials. But my hope is that as some of these drugs work in ALS, that they'll be brought forward to people with PLS. But I'd love to hear Nazem's thoughts on this.
Yeah, no, I fully agree. I as Merit mentioned, I had a special interest in helping people with PLS. Unfortunately, PLS is excluded from most trials that we do, and as Merit mentioned, there are no good models of PLS that we can test drugs on. And it's more rare than ALS is rare. This is more rare, a lot more rarer than ALS. And it's very slowly progressive, which means that if you have a disease that it's very slowly progressive, that means you need, hundreds, maybe thousands of patients in order to see an effect. So combine that with a very rare disease makes it really, really challenging to enroll. I always hope that we can show that something is working in ALS and then just treat people with PLS, assuming it's a motor neuron disease that falls under the same spectrum. A lot of PLS patients, for example, take riluzole and other ALS approved medications.
Thank you for both of you guys' answers, and of course we do hope maybe in the future that it can be applied to PLS, like you were saying. Our next question up here is, how were the US sites chosen, and are you opening more in other states at some point?
I can answer a little bit, but it's done in a couple ways. One is getting site interest, and then looking at the sites that are interested at their quality based on their prior experience. How do they do with enrolling participants and the quality of data that comes from them, and then trying to get a good geographic kind of distribution. We are in a unique time in ALS, which is a fantastic time, which is that there's a lot of trials. So whereas maybe a couple of years ago, every site in the country would have raised their hand to be part of a trial, we are finding that some sites are they're at capacity. So it's kind of a mixture of those kind of factors. I'll defer to Nazem whether they might be opening up additional sites or not.
Yeah, no, these are exactly the factors that we were working with when we are picking the sites, plus kind of the impact of the pandemic and the staffing kind of issue. Some sites also had not only a lot of demand because there's a lot of development in ALS, a lot of clinical trials in ALS, but on the other end, you know, they didn't have enough staff and they couldn't hire enough staff to actually run these trials. So we're doing the best that we can. Currently, these are the kind of the sites that are open. We do have a short list of backup sites in case, you know, enrollment is slow or one of the sites, you know, for whatever reason doesn't, you know, is unable to enroll. You know, as I mentioned, there's so many factors, including staffing, that might, you know, slow things down. So, but these are currently the sites that are opening. I think clinicaltrials.gov is, we try to keep it up to date as much as possible. So keep an eye on clinicaltrials.gov and see, you know, if a site close to you opens. If not, you know, look for the closest site to you, and if you are able to travel, we can, you know, we can try to help as much as possible to make it as easy and seamless for you.
Excellent. We're going to stay on the topic of location. And this is one I think is very interesting. Do the visits for participants need to be at the same site throughout the trial, or can a patient travel from one site to another?
So they have to be at the same site, and both in the double blind and the open label, though sometimes there's some flexibility around the open label. And this is largely driven by a few factors. One is kind of operation. They're all operational, that the drug supply is kind of linked to a certain person, a certain pharmacy, and also the contract and the consent. So I wish there would be a day where it's easy for people to go from one site to another in the trial, but for most trials, it's really operationally very challenging to do it on the back end.
Sorry, Grace, I think we still got you muted, so I'm just going to ask the next one to keep it rolling. Do you have a rough estimate on when full enrollment of 260 will be reached?
So, I mean, the only thing that I can say that I think we have a momentum now. We just have, I think most of the sites, we have 70 sites across the globe, and most of them are now open for enrollment. So I think we do have a momentum for enrollment. I think on the slide, and we didn't prepare these slides too long ago, we had 51 patients, you know, enrolled on when we made these slides, but I can tell you today we have 70 patients enrolled. You know, so you could see how the numbers are, you know, going up kind of quickly, and I hope we can keep that momentum going, and really to find, you know, to really answer the question, does this compound work or not, as quickly as possible. So I would say that we are really working really hard, everyone, to kind of get the enrollment up. And, you know, again, this is something that all of you can help us with by, you know, whether you enrolling or talking to, you know, to your friends and connections and contacts and anyone who's interested in the trial to kind of join.
Perfect, thank you. I'm not muted anymore. So the next question here is, do you think RIPK changes with patients' population or demographics? Do you have any thoughts on this?
First part of the question, sorry, I didn't hear you.
Do you think that RIPK, the RABK, changes with patient population or demographics? And do you guys have any thoughts on this?
Sorry, go ahead, Nazem. Sorry.
Yeah, so I think maybe the best answer is that we don't know if it changes or not. The reason is that you really need brain tissue to kind of measure the activation of RIPK1 in living people, which is impossible, as I mentioned, because we can't do brain biopsies. From what we saw in the postmortem, so the samples that we got from patients who had ALS and passed away from ALS, we did see a variation of the RIPK1 activation in these patients. But that variation could be due to the speed of the disease, could be due to the location where the sample was taken from. For example, if someone had the weakness starting in the arm and they had very severe weakness in the arm when they passed away, if we take that sample from the area in the spine that correspond to the arm, we will see a lot more activation versus if we take in the same patient a sample from the lower part of the spine, we will see maybe less activation. So there is variability. We've seen it in the tissue that we had from patients, but it's and unless we can measure it in tissue in living people over time, we will not be able to answer this. It's a very important question, but I think the best answer is that we don't know.
This has been a wonderful Q&A, and I believe this will be our last question, and I think it's a perfect one to end on. Are you posting the trial information on social media to get word out to recruit patients? And in general, where can individuals go to sign up and learn more?
Yeah, so it's a good question. So there are rules and regulations about postings and advertising for trials. A lot of times we need to get IRB approvals. We have, for most of the sites, we provided some recruitment tools, we call them. There is the website, and maybe I can share the website again. Let me see. This is website on the this is basically on the Sanofi website for the trial. So you can go there and you can find the information there. Actually, it has it asks you a few questions and you can see if you're eligible or not for the trial. So it's kind of a little bit interactive. There is the clinicaltrials.gov also has information and contact information and list of sites, so you can contact the sites themselves. I mean, feel free to contact us also and ask us if you I don't know if our contact information is somewhere here or not. Maybe it's not here, but I'll make it available. So if you have any questions, either me or Dr. Li Shang, who's the medical director leading the study, will be more than happy to help you.
Amazing. And for those of you who are on the call right now, I know Indu has posted in the chat a link to that study, so you can go click it right there. I want to thank both Dr. Cudkowicz and Dr. Atassi for joining us tonight. This was a wonderful presentation. We know you are very, very busy individuals. The next portion, you're more than welcome to stay for. If you have to leave, we understand. It will be our non-recorded open forum where we allow everyone to speak on whatever they need to speak on. So I want to thank you again very much.